Okay, so in this video, I'm going to be talking about meiosis. So meiosis occurs as a replication of diploid into haploid cells. So basically, this happens in sex cells rather than autosomal and occurs in two phases. So your first phase is M1 or meiosis 1, where the diploid turns into a haploid. Then the second phase is meiosis 2, where the chromosomes split. Kind of like that. So just like mitosis, this really starts to begin in interphase during the S phase. So I'm gonna move this a little closer. So your S phase, you have your nuclear envelope here. This whole thing is the nucleus. Here in your nucleolus, you have spindly chromosomes that start to replicate. They don't become chromosomes until prophase 1? Yeah. But for continuity's sake, let's say we have these pairings of chromosomal information. So this is the S phase. This happens during interphases and technically part of meiosis. Now, the first actual proper phase of meiosis is the prophase. So here you have your diploid cell. Basically, the nuclear envelope, the main part, the most important part, is that your nuclear envelope fades away and your microtubules start to develop out of the centromeres or centrosomes, and your chromatids line into chromosomes proper, kind of like that. I'm going to draw one more for continuity. This is prophase 1. Next is your metaphase 1. So this is where things really start to become different from mitosis, because instead of the chromatids themselves splitting, basically you have these pairs of chromosomes called tetrads splitting. And we'll draw it out to explain it a lot better. So here you have two pairs of chromosomes. I'm not going to draw them in super detail, so you're going to have to use a little imagination here. But you have these two different pairs. I'm just going to box them out that are connected to your spindle fibers, like this. Now, this is a major part of meiosis because here you have what's known as crossing over. So these little butterfly guys are your chromosomes, and there's little points where they can touch during metaphase 2. I forgot to write that down. Or metaphase 1, excuse me, where genetic recombination happens, called crossing over, where alleles switch in places where they aren't necessarily supposed to. causing a genetic shuffle, if you will. And this can create new genetic information being created. So now, onwards with our separate path for meiosis, you have your next phase, which is anaphase 1. Where the cell starts to stretch a little, becomes a little ovular. And instead of the chromatids being split, your tetrad pairs become split. Now, imagine for yourself that a crossing over happened. I just don't have any extra colors to really go into detail with that, but they did. Now, something I forgot to mention, though, is that this information is paternal and maternal. Some of it comes from your father, some of it comes from your mother. The homologous pairs do, at least. And they don't have to be in any specific order. Some of them kind of pair at, not necessarily random, but in a planned, I can't think of the word, a planned separation, I guess, is probably a good way to explain it. So here you have your anaphase 1, where the cell starts to stretch and the tetrad pairs split instead of the chromatids. This then moves you directly into telophase 1, which is very similar to in mitosis. However, you now have half of the genetic information you did before. Here you see the little cleavage furrow. And you have your chromosomes. Now, here you start out with a diploid cell. You have that di, meaning 2. Your diploid. Now you have 2 haploid cells. I don't honestly know what HAP stands for, but this is telophase 1. You have cytokinesis happening, very similar to how you did in mitosis. You have that peanut-shaped kind of dual cell that's starting to split and cinch. And afterwards, you have 2 new haploid cells. So this is interphase 2. It's very brief in the cell's life cycle, at least the gamete. Now you have 2 cells with a split of the genetic information of the original cells. Interphase 2. Now from there, you go through meiosis all over again, essentially, but a little more brief. It's basically mitosis, but with your new haploid cells instead of your diploid cells. So your new shuffled chromosomes will start to phase back out, and your nuclear envelope, in interphase 2, the nuclear envelope has reformed. That happens in telophase 1. But in prophase 2, we call this P2, your envelope starts to fade away again, and very similar to in mitosis, the centromeres create their spindle fibers, and the spindle starts to form and does its thing into metaphase 2, where now you have, again, just like in mitosis, your haploid information being lined up like this. It's a little more simple, but it's just because there's less genetic information. They line up. This is metaphase 2. And then your anaphase 2, where they split. Again, very similar to mitosis. And then telophase 2. Now you have 2 peanuts. Instead of 1. There, telophase 2. And that's how you end up with 4. Oh, there we go. 4 gametes. And that is the 2 phases of mitosis. Or no, excuse me, meiosis.